Welcome to another episode of the Being and Doing podcast, where I'm interested in the curious and interesting minds that are all around us and bringing you their stories, which will hopefully inspire and maybe sparkle a bit of investigation in your own being. And today I'm really privileged to be joined by Dr. Catherine Green So I normally start this podcast by saying what's my impression of my guest, but I can't start without saying Catherine was one of the person in the Oxford AstraZeneca team who developed the COVID vaccine. So I guess that's the first thing that people would say. But I think what was my impression when I heard you speaking on the Genome Stability meeting, on the conference we were in together, was just the down-to-earth presence and also just sparks of curiosity that I could see in you, and also just really genuine interest in science. And I was inspired, being someone who is a scientist, to observe that in you. So thank you for the privilege to have this conversation.
Thank you. Those are very kind words. I will take that.
So as I mentioned, the first questions often is, what are some words that you identified with, either values that you hold or some identities that you feel really comfortable in?
It's a really challenging question, that I'm not sure about. So when people meet me, they would say that I'm lively and I'm loud. And I do hold that as part of my identity. It's not always common for scientists to be exuberant, perhaps. And I think I do have that. And I think that's an asset. I think it's been helpful in this part of my life and in others to be lively and exuberant and loud. And I hope in that also welcoming to ideas and to people and to events. And perhaps that ties into where we're at today.
And actually, that's an interesting thing about what we have as an image of a scientist.
Yeah.
And maybe we could explore a bit of that, because I haven't spoke about that with anyone, because I guess the image is not too feminine, but not too this, or not too lively, like very composed and rational. And so how difficult or easy was for you to accept your liveliness when maybe it has been something that stands out in the crowd of scientists?
Yeah, so there is perhaps some pressure to quieten that aspect of me down, but it is an intrinsic part. I think if you ask my friends, I've just got a loud voice. I'm a loud person, and so it's a conscious decision perhaps not to try to suppress that. And that perhaps helps in some ways, because you get noticed, and science is sometimes about being noticed. But of course, it does mean that if you're wrong, and scientists, of course, are always wrong, that also gets noticed. So you have to be able to be prepared to take the knocks that come with being noticed. And so perhaps if you're going to be loud, you need a thicker skin, but that's okay. I've developed that over the years. Those two go hand in hand. And this idea that there is a perception of scientists and how scientists are, because of course no two scientists are the same. There isn't one.
No.
We're not one amorphous mass in the same way women are not one amorphous mass. But that is still quite true in the perception of scientists outside of science, or particularly in media presentation stereotypically of scientists. That still pervades, doesn't it? That we're serious, because we do a serious job. But you can do a serious job and not necessarily have to be serious about it all the time.
Yeah.
In fact, doing a serious job sometimes requires a bit of lightheartedness to get you through the seriousness, perhaps.
Yeah, I can totally relate to that.
You have to be silly sometimes,
Yeah.
And sometimes science is boring, and so it's okay to be silly about it.
Yeah, that's something that maybe we could again talk about, because often I guess that there is that perception that what we're doing is quite elegant and neat, where actually a lot of our work is sitting in a dark room for hours, or I don't know, in the vaccine production. But I can imagine there is a lot of being dressed in like suits that are really not comfortable, and things like that. So maybe you can say to people how day-to-day work of a scientist in working in vaccine production.
Well, that's right. Well, as scientists in any subject, a lot of it's repetitive. A lot of it is repetitive on the day-to-day level when you're at the bench, which I don't get to do so much anymore. There's a lot of moving invisible amounts of clear liquids from plastic tube to plastic tube. It's very boring manual work, but yet has to be done with precision and accuracy, and so you have to concentrate on it. So there's a lot of very boring, dull stuff in order to get to a successful and interpretable experiment that people don't realize. And perhaps that's part of the nerdiness of the projected image, that you do have to sometimes just concentrate on doing the thing. But of course, then you have to, once you get that exciting result, you then of course have to go back and do it again and expect the same result, and then you have to go back and do it again. So there's a lot of boredom and repetition that perhaps is sometimes forgotten. And I need some tunes to get me through that sometimes, or I need to be able to surround myself with interesting people to see during my coffee break, because I know I've got to go back in there and do it all again and expect the same result. So there needs to be a bit of spark, and we need that passion. And all scientists, even the quiet ones, the ones that stick with it and do it because they love it, have that passion. And I've learned to appreciate, as I get older and as I work with more and different and diverse teams, that even my quieter colleagues have that inside them, that passion. It's just we do of course express it in different ways, and I'm a jump up and down and swear a lot. This is effing brilliant, but others are expressing their passion and their joy for what they do in different ways, and we have to learn to recognize that in others. One of the things about being a leader is allowing all of those voices to be heard, both in happiness and in complaint.
And that's interesting, as you're saying that, people again have this perception that science is there is a eureka moment in science. And I mean, I can imagine for you getting the results that the vaccine works may have been one of them. But I know for me, I just don't allow the eureka moments to myself, because you're like, wait. The next replicate has to come in for me to be kind of allowing myself, and then the moment passes.
That's right, isn't it? That's right. There's that voice in your head like, yes, I've got it, when you get the perfect Western blot that shows the phosphorylation. When you get that, you've been trying to set this damn experiment up and you get there and it does, but then there is always in the back of your mind hang on, might be a fluke, I've got to do it again. So by the time you've got it 3-4 times and you know it's good, you're bored of it and it's just another figure for the paper. And that's a shame, but it is true, we have to. And that's again, it's one of the worst things about being a boss, is your student will come into you with the result, look, it's worked, and I'm like, well, it's nice, but please do it again. That's a shame, isn't it? And also that a lot of science is cumulative, and that it builds to the thing that is the idea. It builds to the thing that is the overall result, and there isn't always one eureka moment on the way. You look back at it and it's the cumulative. Oh yeah, that was interesting. That was interesting. Oh, this is interesting. And with the vaccine, there were a couple, and of course, as with all things in clinical trials in particular, there's a lot of waiting around, and it's a relatively new field to me. I've only been doing manufacturing research for 5-6 years now, but there's a lot of waiting around, because by design clinical trials require that you, for us, immunize participants in a trial, and then you have to wait, and so the eureka moment is tempered by the fact that a result will come, you don't know when it will come, and it will be a long time after you've, for us, done the manufacturing, because we do the very early parts here. We make the vaccine, we hand it over just to the clinicians and the trial teams that then go on and run the trials. So we actually hear the results we're the very last people to hear about it. And so for us, there was one moment in the process where we had the first clone, because the vaccine is a cloned virus particle, and we had a clone first growing. So that's microscopy, looking down the microscope and seeing if we get plaques effectively on a bed of cells. So we knew we were starting to see vaccines come. But at that point, although that's great, that's an in process, we're going to be able to make a thing. You at that point have no idea if that thing will be effective. So although you have those kind of moments through the process of, we can do this, whether this is any good doesn't come until much later. So it was a strange year of highs and then really not knowing if what we were doing was going to be of any value to anybody, really, until right at the end of 2020. It was a year of waiting to know if it was going to work.
And that is something, I guess. I haven't said this about me, but I'm training as a psychotherapist as well, and so there is this moment of being able to sit with the unknown and to be comfortable in the uncomfortable. And I think science really prepares you well to do that. And I'm just wondering, along your career, what was your way or process to develop that sitting with the unknown and allowing results to accumulate without being too attached to the outcome?
Yeah, it's a very good description of the process, isn't it? Because sometimes projects do take years of persistent accumulation until you understand what those first results are. Often the first results are just something peculiar or unusual, or that doesn't look quite as I expected it to look. Maybe there is more to uncover here, isn't it? And that's some of the joy of it. Is the sitting with the. I love the fact that we don't know things, and presumably that's something that all scientists have in common, because we go into this because we don't know stuff. And if everything was known, there's no point in us. So you have to be able always to have joy in the not knowing of stuff, which is the one that's hard to explain to the children. Kids, when you go and speak in schools, expect scientists to know things. And I'm like, well, the whole point of being a scientist is really knowing what we don't know, because knowing what we know, that's the unoriginal part of it. So I think, and yet you need to know a lot in order to know where that space is that we don't know. So I suspect that's a common trait, that this ability to be comfortable with not knowing stuff, and the fact that that is the brilliance of it, because you can work in that void. And even if it's minor things, I mean, sometimes, so prior to doing vaccine work, I worked in genome stability, and sometimes it's really, there's this polymerase and nobody really knows what it's for, and yet it is conserved, so it must do something. So I'll just poke around that a bit. Or, this protein has a domain of unknown function, but it's there across mammals. Maybe it does, what we just poke it a bit. And I hope that we don't lose the ability to do some science like that. The kind of exploratory, I've noticed a thing and I just want to have a go at it. And it gets much harder to do with our funding regimes being very targeted towards, can we translate this into a benefit for mankind? And sometimes it's more esoteric than that, isn't it? We don't know can't write in a grant proposal why this is interesting, apart from I just want to have a play. And I hope we don't always lose the ability, because I think when I started my PhD, we were able to play around a bit more, perhaps.
Yeah, that is something I have had discussions with scientists, how limiting to actually original science it is, that we need to write outcomes, and obviously it is challenging with a lot of scientists being in the game and then having to disseminate means to do science. But I wonder, I spoke to someone at the Crick who is a philosopher, and we were talking, it would be really great if people would leave 20% to chance and just randomly allocate means. 20% is really just randomly allocated, just to leave that place that some things will come out, not because of prior record, but because people have that curiosity that then will drive. So actually, I think I feel like that genuine curiosity is there with you. So how did you come to be a scientist, actually?
So I took a very linear path, really, although it probably wasn't the path I intended to take. I think if you ask my mum, I was always just interested in things. When I was a child, I did like to turn over rocks and see what was underneath them. And she would say, if I'm sat in the bath as a young child with the things that you pour water through, wasn't really playing with those. I was doing experiments with them. I think she was, quite focused on where does the water go and how does it go like that. So I think science for me at school was the only subject that I was interested in, just because I think I was always I'm very happy to not know things and to ask the why, which presumably made me an enormously annoying 8-year-old, but I just think I was a nerdy 8-year-old and science seemed like the place for me. But then when we're looking for university, I think because I mean I knew I needed to get a job, and I didn't really know scientist was a job as it is. So I went to Cambridge thinking I would study chemistry, because in North Kent, where I'm from, there were pharmaceutical industries. Chemist was a job, not pharmacists, chemists, people that make things from molecules. So I thought I'd go to Cambridge to be a chemist, and then when I got to study chemistry at Cambridge, you can't just do chemistry. You do this degree called natural science. It's the same now. And so you have to take modules from other scientific disciplines. And so I took biochemistry, or cells, it was called. But it introduced me to this concept of biochemistry and biological systems as chemical, complicated chemical environments. And from then on, I was hooked on biochem, and that took me to genetics, and that took me to this molecular biology, we'd call it today. So I think it was that. I think it was both the knowledge that I think I was something in my brain was encouraging me to do that questioning, and then I discovered that this biology of cells existed, and I thought that was extraordinary. The fact that we could perhaps break down biological process into pathways and chemical understanding of nature was fascinating to me, and I was hooked on that ever since.
I have a big smile because my journey is quite similar. I mean, I was really interested in biology, but then I went to a science school where I started to fall in love with chemistry, and just this idea of how chemistry can help us understand the complexity of biological systems. So I did also end up studying biochemistry.
Yeah.
So,
and then during my PhD was the years when we were starting to get, so we got the first full genetic sequence of the Saccharomyces cerevisiae, and I was working with a professor. It was a Cancer Research UK. They were called Imperial Cancer Research Fund then. I was working with a guy called Noel Lowndes, and we were studying DNA damage checkpoints in cerevisiae. And we just had the full genome sequence of cerevisiae. I mean, it was an absolute game-changing experience. You could have it. We all had these ancient MacBooks because it's the dark ages, but the idea that you would have a whole genome of an entire organism, like every gene in the yeast was there for us to interrogate just by searching, was just the most extraordinary feeling. And that it was a very interesting and lively time. This was probably late 90s in Clare Hall Laboratories, which I don't know if you've ever been. It's like in a lorry park at the outskirts of the M25, a really odd place. But it was a very intense place to do science, because once you were there, you couldn't leave. There's like a pub in the village, but that's all. So you got to work, you stayed at work. And I was surrounded by these fantastically interested scientists. So Tim Hunt was down the corridor, Thomas Lindell was down the corridor. Jesper Sveistrup is in the building with you now, I think. Rick Wood. I mean, it was just the most, John Diffley, who you obviously all know. So we were all there in this field in North London, surrounded by rabbits, with nothing to do but talk about DNA and genomes and genetics. And we could start to have these tools that were new. It was just the most extraordinary time. And so I loved being a PhD student there. We worked really hard, but it triggered in me this idea that you can be just interested in a big world of science, as well as focusing on a relatively small question, because you're going to focus in on your one thing. But we used to talk in the canteen about building a synthetic genome. Can we build an organism from the Sigma catalogue? Was always like, we were very, very nerdy place to do really intense science, and I loved it. And that after that PhD, I was like, this is obviously what I want to do, very naively, because I didn't know how difficult it is to succeed in science. I don't know if I succeed, but I've managed to keep doing it all these years. So that gives me great pleasure.
And actually, maybe so part of this podcast is I asked what success is for you, and maybe this is a point to define it, because it is very difficult to talk about success in science, especially because it's very hard to measure. as you said, it's very cumulative, and then eventually one person is on the precipitating point, but there are so many people coming before that it's very difficult to distinguish who actually made the success. So what is it for you now?
Yeah, I always think about the people that I've worked with, and hopefully people who have come behind me who then go on to do good things. I've always tried to have undergraduate students, interns, to give people the opportunity to come into my lab and have a go, and to see what it's like to be a scientist, and then to go out into the world with that experience and try to make something of themselves. I had the opportunity to have some great bosses in my life, and people gave me opportunities. And so I always try to, those people who I see as being interested, but perhaps who wouldn't otherwise have been encouraged. So for me, success is encouraging others, whether that's through lecturing or teaching or bringing people into the lab, or being part of graduate training programs, all those backroom things that we do as academics. I think those are the only true successes, because having a success for yourself is only ever going to be small, but you can do a lot by encouraging others to be your legacy of ideas. I just want to, if people would just have the bravery to follow their own investigative mind, to question an idea, to put a hand up in a lecture. If one kid that I've spoken to puts their hand up and says, "But please, miss, I didn't understand this," then that's success, isn't it? Getting the next generation to carry the torch forward.
I must say I'm very touched by this, because it's also something that drives me as well. And especially in this day and age where it is quite difficult to stay in the field. Building a generation of inquisitive new leaders is really important. And I'm curious, you mentioned leadership, and what changed for you going from the bench to leading? It's a completely different job, I think.
It is and again, it's one of those things, and this must be true in all careers, that you start off by being good at a thing, and then you get promoted out of doing that thing to doing something at which you have no skills or expertise, or sometimes any desire to do them. But I do like it. I feel comfortable in that, and I know that others don't so much, but I actually, as a bench scientist, I mean, I've got quite good hands, but I'm not very good at the repetition. I'm not very good at the having to do the things lots of times. I'm a starter, not a finisher. I acknowledge this by the time I get to my age in life. I'm the ideas person. I like to get things going, and then the finishing of it is awful. So I have to surround myself with people that are really good finishers, and so that's something that I find I'm much more comfortable in. Building teams now that work together is something that I actually think I just am actually quite good at, and you don't know that until you start doing it. It's weird, isn't it? You get put in this position where you have to try these new skills, and then so I enjoy that very much. I enjoy that aspect of working in teams, and I always did enjoy working in teams, even when I'm a part of the team rather than the boss of the team. So for me, that's one of the great joys of science, which is always, again, it comes down to that idea of how sciences are perceived by others, is that I have never worked in a team where one person is the standalone person. It's always the team that does the thing, and so recognizing that and trying to build those great teams that can be more than any one individual would achieve is another great mark of success, I think. And I think you do see that in the good scientists that you surround yourself with. They generally, what they do is they build good teams. Perhaps you can be a superstar postdoc for a bit, but if you're going to be a PI and be successful, you've got to keep finding good combinations of people to get around you. And I'm actually not very, I'm quite a good delegator. I'm quite happy to let other people's ideas be better than mine. And that's another thing that so leadership, it's an odd thing, isn't it? But for me, it felt like a natural progression of me just being a bossy loudmouth. I don't know. I think it comes to that. Yeah, I like that. I like people. So the bringing together of teams is something that I just naturally like to do.
And what might have been a challenge for you in that position?
So I had to learn very early on, I got some good advice, and I'm trying to think where I got it from. I actually went on one of these junior PI leadership courses. I don't. It was run by Embo or
Yeah, I
something.
just came from that one. It's
Oh right,
amazing.
yeah, and
Yeah.
it really. I mean, a lot of it is obvious, but you don't necessarily clarify it in your brain as a strategy or as a thing you really need to think about. And for me, it was this idea that you have to make space for quiet individuals to be heard. So there have to be multiple routes. As a leader, there have to be multiple routes that people can communicate with you and you will hear them. That not everybody is going to speak up in a lab meeting situation. That there will be those who are comfortable in talking in those situations, but there will always be individuals who are listening in those situations and taking it in and formulating ideas, but not necessarily vocalizing them immediately. And I am a person who thinks by talking out loud. You can probably hear this. I haven't had these questions in advance, so I'm just making this up as I go along. Yeah, I'm thinking out loud. And that's fine, but there are also always in your groups people who think in their heads and then come out vocally with very well thought out statements that have taken them a lot of time. And so for me, all the things that I say, I don't mind if people disagree with them or say that's a nonsense idea, Kath. I'll be like, why don't we try knocking out this gene because it might do that? And somebody will say, stupid idea, Kath, because A, it's already been done, and B, it was a stupid idea when they did it. You know? And that's fine because I'm just thinking it. It's not an action. I'm not telling anybody to do it. And yet other people tend to be quieter in meetings. When they say a thing, they've really thought it through. And if you just immediately disregard that because you think it's daft, that's hurtful to those people. So it was a lot about learning that not everybody thinks and works their brain and their communication style the same as I do. And I have to find space to allow everybody's voices to be heard and given the appropriate weight for the amount of thought that's gone into it. And that was really insightful and made my teams better because I really do try to say, right, we'll talk this in a meeting, but if that's not your style, send me an email. If that's not your style, call me. We'll set up a Teams meeting, have times where different means of communication are available to everybody. It's like you have to make space.
Yeah. Thank you for that. Yeah, it's something that I really appreciate you have an awareness of because it is challenging, I guess, to keep different polarities of being in a team. Actually, you spoke of being involved in genome stability, interested in polymerases, and then on the other hand, you are working in clinical trials and getting drugs to the market or vaccines to the market, and I'm just wondering how challenging was that? Because for me, that was quite amazing to see the gumption, the risk taking that I imagine maybe it wasn't like that for you, but I imagine it needed to accept position like that. And then you said it was only 5 years back, which means that during COVID, you were there for a year or two, which is even more of like more admirable than to know that you can take those risks.
So yeah, it's a route through that made sense at the time, I guess, is always so as you know, so trying to keep bringing in grant income to support your salary is the biggest challenge of being a PI, particularly in the academic sector. We tend not to have, unless you get, until you get your lifetime chair, which are extremely rare, and I don't have one of those. You have to keep bringing in grant income in order to fund not only your research, your team's salaries, your PhD students, your consumables, also your own salary. And so part of the way of doing that is to make yourself useful to your institution. Yeah. So you say yes to the administrative and organizational jobs that a university needs. And so, as well as running a research lab at the Wellcome Centre for Human Genetics, I'm a person who gets stuff done. I'm vocal on committees. I can stand up for things or for facilities. So I was helping to run a couple of the core services that exist in that Wellcome Centre. So I was running Chromosome Dynamics, so that's karyotyping or microscopic analysis of genetic material, and helping support their microscopy core and their fax core, because those are equipment that I know how to use, and so I can be the kind of scientific and the leadership and the kind of management structure on top of those research services. So I was running a research team and doing some core research facility management, and then the facility that I now run, the clinical biomanufacturing facility, was their head had retired and they hadn't found anybody to replace it. I think the department didn't really know what it was. As you can imagine, running what is effectively a manufacturing factory, and licensed MHRA approved manufacturing factory is quite a unique thing. Yeah. And so others in the department knew it existed, but to really justify what it is, why it's there, particularly because it was tending just to make vaccines, so quite focused on working with scientists in the Jenner Institute, one particular part of the University of Oxford. So it hadn't had a head for a while, and so there was some financial, it was just, it needed somebody to go in and look at it. And because I was managing other facilities that run on the same kind of system, and because I'm noisy and people know who I am, the head of department had said, Kath, will you just go in for 6 months and just take a look at this, sort out their finances, and then we'll sort out how it needs to be restructured and we'll go on and take it from there. So it was really like Richard just dropped me in. I actually thought he was going to sack me. When you get an email from the head of department saying, Kath, will you come to my office this afternoon? I'm like, oh my God, I've done something terrible. But he was going to offer me a job. He was going to say, go and do this job. So I went in for that 6 months to kind of meet them all and find out what they do. And it was very clear to me that the day that I walked in, that this was a team of highly dedicated and hardworking, really well, extremely trained, very specialized scientists doing something that almost no other UK university has. So it was immediately obvious to me that this facility is something really special, doing something really unique, because getting new drugs to clinical trials is a huge challenge and it's hugely expensive, and being able to do that in-house at cost and take on some difficult projects, it was clear this was an asset to the university and something that needed to be built back up and nurtured. So that was my kind of role. And it turns out that making biological medicines is just cell culture. Yeah, it's either growing Saccharomyces or Pichia or E. coli, things I know how to do from being a PhD student and a postdoc in a molecular biology lab. Yeah, so it's just the same kind of expertise. So I'm a biochemist. I know how to purify proteins. I know how to culture cells. I know how to do transfection. So the scientific skills, I had those. I didn't know that I had them, but I did have them, because it's just doing science. It's just bigger scale than doing it in the research laboratory, but it's the same techniques and the same understanding of analytics and assays and of quality. And then it's just an extra level up because of the complexity of doing it in a compliant fashion. So there's a lot more paperwork, which is not my area of expertise. And there's the necessity to abide by the regulations, which is not my area of expertise. But it's clear within the team, they have fantastic people who can teach me at the same time as keeping those things running. So it was, as soon as I got in there, I think, within the first week, it was very clear to me that this was something that I was going to do, and that I was not going to let Richard hand this over to somebody else at the end of this 6 months that I was getting parachuted. I think I can work with these people, and I think we can together do something really good. And so since going in there, 2018 has been my mission to keep that running, to take on more, to expand what we're going to make, to move with the times, to make sure that we're able to make the kind of medicines that the scientists within Oxford University are going to invent over the next 5 years. So biologics, gene therapies, cell therapies, new vaccines, new therapeutic modalities, monoclonals, whatever new biologic a University of Oxford academic wants to make, they should be able to come to me, and my team can advise them how to start to make that into a process which will be suitable for GMP, which is the procedures that we have to follow to put something into a person, or we can advise them, how to get somebody else to make it. But to have that within the university, I think makes us quite, it was shown in COVID, makes us contenders for being able to respond to big events in the world, but also for just generally pushing forward where the landscape of new medical products can be, because we can do new things because we're scientists as well as being manufacturers. So we can take an idea and perhaps do something new in a creative way. And it's been a revelation to me that it's something I didn't know I liked, but I actually it turns out really like making a thing.
Yeah.
Because scientists make ideas, yeah? So I've
Yeah.
been used to, we have a concept, we've got a new interaction, we've got a new kinase, a new idea in the world in a paper. But I actually really like making a physical thing, because we make a vial of medicine that we then ship to a clinical trial and it gets injected into somebody's arm. And there's a huge pleasure in that, and that we've made this. Every time we do a fill, we all get together, we have a little celebration. We're like, yeah, we've put the medicine in the vials, and it's hugely satisfying to make a thing. Maybe I should have been an architect or an engineer. When I look back at my life now, I think I should have built motorway embankments or something. I should have made things, not ideas. And now I get to do that. You never know where your career will take you, because now I get to make actual things.
Yeah, and I guess what I'm impressed by is actually your astute ability to value people and potential, which I think
it was clear to me, and I still see that in my team today. They are a fantastic. They shout out to the CBF. They are a fantastic bunch of people. They work really hard, but they're hugely knowledgeable and very niche.
Yeah.
And yet not afraid to take that very targeted knowledge and apply it to a completely new question. How do we make this new thing that the scientist wants to make that's never been made before? Yeah, we're always going to make safely, so that we are comfortable enough to inject it into a participant volunteer in a trial, yeah, and that the regulators will approve that. How do we make something for the first time safely? And that they are a fantastically driven, interested and interesting bunch of people. And without me walking in the door and having those people around me, I wouldn't have kept the job, would I? I've done it for 6 months, handed it back to the department in a slightly better state. And then who knows where we would have been, because that was 2018. So when 2020 hit, the beginning of 2020, the first week of 2020, when Sarah had called us and said, I think we should worry a little bit about this virus in China. I'm a yes person, yeah. So part of me pushing the CBF to do new things is me just saying yes to everything and then sorting out how quite we're going to manage that. So the challenge was, how fast can you go? How quickly could we have material ready from Sarah's idea to get into the arm of a person? And I'm like, oh, yeah, I think we can. We can do it much faster than we've ever done it before. And we can do that at risk, but it's at financial risk. Yeah, we're basically going to do the thing and hope somebody gives us some money afterwards. We're going to ask for forgiveness, not permission, for maybe slightly reorganizing the structure of things, put some projects on hold that, shouldn't really be on hold, because we need those vaccines too. But actually, we're going to try and respond to this as quickly as possible. So the ability to just be brave enough to just say, well, let's go for this, because if we need it, we're really going to need it. And if it turns out to be a waste of time because this just fizzles out, then I'll go and I'll make those apologies and say, look, we made a call and it was the wrong call. And so all your projects are 3 months delayed, and I'm sorry about that. But I didn't have to make that call, because obviously the decision to go fast and go early was the right one in the circumstances.
And that's interesting, I think, in terms of leadership, the importance of being able to, you mentioned this already, I say something, but then I'm able to also deal with the consequences of it. And I guess it's really important to be able to take one for the team as well when things go wrong. And how difficult or easy was that for you?
So I do. That is my only job. If I'm going to push the team to do a thing, then it's my response. The buck always has to stop with me. Yeah. So that is my only job. I will fight their corner, and I will acknowledge that if a mistake is made, at the end of the day, that comes down to me. That is always my job. And then with the COVID vaccine project, actually, it wasn't quite like that, because there were 6 of us. So very early on, it became clear that in order to go faster than ever been done before with a vaccine project, what you have to do is you have to do all of the steps that you would normally do in linear fashion. You have to start them all at once. You have to do them all in parallel. Yeah. So while Sarah's doing the vaccine design, I'm already starting to prepare the clean rooms for a manufacturer. And so we've got Sarah Gilbert, myself, we've got Teresa Lamb, who's our immunologist, fantastic scientist, going to do all of the immunology and all of the animal studies that are going to be needed to show that this vaccine is working, and then do all the immunology on the trial participants to show that they're making the right antibodies, they're making T cells. So a huge immunology project sits around doing a clinical trial. And so she needs to get started straight away planning for that, planning for 40,000 samples coming in from a clinical trial. Yeah. So she's in the room from the very beginning. And then Andy Pollard, who's going to be the lead clinician on that, running the clinical trial, he's in the room from the beginning. Adrian Hill, who's head of the JENA, who knows everything there is to know about making vaccines and translating them to large scale and getting them out because of his malaria work. And then Sandy Douglas, another PI, who's really interested in process development, how you make lots of vaccine really cost effectively. So really early on, from the very first weeks of January, there were 6 PIs around the table making decisions about the project as a whole, but also giving input into each individual step of the project. So it was never just one person. So we talked a lot, the 6 of us, in the very early stages. And because we were knowing everything was so you always had a sounding board. You always had this collective, somebody to check, is this crazy? And so it meant that you always had somebody to go, yeah, it's crazy, and we're going to do it anyway. Yeah. So it was not just a sounding board. It also was a motivator. Between the 6 of us, we have everything we need to do this as quickly as it could possibly be done, because we've already planning for the end at the beginning. And all the expertise is in that room to do the things that we need to do. So it gave us, I think, a confidence that you might not have if you're just by your own doing your own bit. But all 6 of us there, a confidence, a belief in ourselves, perhaps. Damn it, we might just do this, because and that impetus to, right, I'm going to say I'm going to do this. I have to damn well deliver it, because I've said it to these 5 other people and they need me to deliver it. So there was no working in a vacuum at all.
Which again speaks to the importance of teams.
Yeah, it was an extraordinary team to be in. I don't know how often you would get, so the 6 of us had all worked together in various different ways, but never that intensely as one unit to do a thing. And it was really eye-opening to me, actually, the benefit of having other senior people around to bounce ideas off and to validate decisions, to quickly make good decisions rather than to quickly make bad decisions. Because I think the decisions that we made, because they were kind of sense checked in the room as we were making them, were really good decisions. We made really good decisions through that process. That's why we were able to go so fast. And it was that collective responsibility and the collective brain. And actually, when I think back at it now, because you reminded me of when I was doing my PhD at Clare Hall, and that community of scientists who were very senior and very knowledgeable, but actually were always, to some extent, working together, because we were working on this really closely related field of genome stability at Clare Hall in the late 90s. And it felt a bit like that, I think, that momentum of we're all trying to answer the same big question. Something about getting groups of people, scientists together who are interested in similar things. There's something very synergistic about that, perhaps.
And then I think it's part of the book, but also I think there is this idea of bad pharma and all these kind of things. And I mean, like, as a scientist, I know that there are amazing things we can do, and then there are logistical things that are just beyond our means, which I think pharma has developed. And I wonder, did you have that distance before you actually joined AstraZeneca, and did something joining them in this project then helped you to work through some prejudices?
So I don't know if we learned much about big pharma, as in, yeah, you're right, this prejudice that we have about them. Some of that is going to be true, yeah. Profit margins can be high. Equitable access to medicines is a challenge around the world. You know, I'm not saying that big pharma are angels.
Yes.
We worked with a very specific team at AstraZeneca. I think within that core 6 of us in January, February, and March almost thought we could do it all ourselves, because we were very much like, we're going to do this, we're going to solve this problem. And then when you actually get to thinking about, hang on a minute, if we're talking about global deployment even if we're talking about doing a clinical trial in 40,000 individuals, that's a lot for a university academic team to manage. But if you start thinking about millions and then billions of doses, we don't know how to fill. I talked about putting the thing into the vials. Yeah, we don't know how to fill 1 million doses of vaccine. We don't know which factory does that, and we don't know how to store that or ship that or move that. So the logistics of it became very clear that we're going to need a partner. And some people at the university talked some sense to us and said, no, we're going to go and find you a partner because you can't do this by yourself. And so working with AstraZeneca was really eye-opening, just for the ability that they have to do some of that. They know how to move stuff around the world, and they have teams in place and systems in place for tracking and monitoring things that we just don't comprehend, but of course you need, and for running large-scale clinical trials, managing the data, just shipments and packages and deliveries and cold chain, very complex. And so they just have teams in place that know how to do that. And so we didn't have to reinvent that wheel. And it was hugely transformative for us to work with teams that just get in and get on with it. I guess in the same way as our team were just getting in and getting on with the things that we know how to do.
Yeah.
We obviously got the experts on board and they just got on with the things that they knew how to do. And there was a bit of, and we're still really good friends with quite a lot of the Astra team, because obviously we worked really intensely together over that year. And so we write grant applications together, we sit on vaccine panels together, we see each other quite a lot. Some tempting things about going, they do good work. I know it's weird, isn't it? We learned that they're just scientists like the rest of us. They're not some evil core big pharma scientists, are they? They've come through, they've done PhDs and postdocs just like we have, and then through different life decisions have gone to work with big pharma companies. But they're still really interested in the why of the science and doing the science behind it. It ends up making money for people instead of ideas, but I guess that's okay, isn't it? It's just a different view of the world. So we enjoyed working with them. We couldn't have done it without them. They don't get enough credit for taking on a project for minimal profit when other companies were clearly going to go off and make large sums of money from COVID vaccines. And Astra were really prepared to do it at no cost in the initial stages and at a minimal project to the end, minimal profit towards the end of the project. So they should get more credit for that than they do, I think.
And that's another thing about, I guess, credit, but I don't think it relates, but I guess it's the public view. And I had moments where, I mean, I know your story of sitting next to someone and saying, well, I know what's in that vaccine, but even though I didn't fully know, I just felt like, again, that there was part of me always saying, do you just know what it goes into making this? And then having that idea that like people don't know what they're doing when they're making vaccines, and it's slightly enraging and also understandable in a kind of field of fear. But I wonder, what was that next step where you, as a scientist who was like just interested about the work and doing the best work you can do, then suddenly need to communicate that to a huge audience? This was not
and so very early on we were involved. Sarah in particular, Sarah started off, she was on the Andrew Marr show a lot on the weekends, because people were really interested in how the vaccine project was progressing and what we were up to, and of course rightly so. And it's a really big challenge, because the science behind an adenovirus vector vaccine is complicated. It is. It's theoretically complicated, and therefore the regulations that we have to go through to demonstrate that what we're making is as safe as it can be and is the right thing, it's really hard. We have to do a lot of tests which have complicated names. We have to try and explain, viral vector delivery mechanisms, and it's not easy to a person who doesn't know what RNA is, or doesn't know what a vector is, or doesn't know that you can have an inactive virus which can be infectious but not harmful. So there were lots of complicated things that we had to start to explain to not just a patient who's suffering from a disease, but to the whole population who aren't necessarily unwell. This is the unique thing, of course, about a vaccine, is it's a medicine that you give to a person who is not sick. And so you're making an intervention to prevent an illness, and it's a completely different ballpark if you're talking about giving a medicine to somebody who is suffering from a disease. The risks that a person is prepared to take in order to cure a disease that they can feel that they have are, of course, completely different, and the way that you have to approach the description of this is completely different. But we didn't really realize that in the early stages, so we're just trying to at first describe the science, and obviously the way that scientists describe science is very measured and very precise, because you only know within the constraints of the experiment that you've done. And we're happy with uncertainty and extrapolation, but we always say, and we take this case that we actually have understood, and from there we extrapolate to other events, but we haven't proven that because it's an extrapolation. And we would always be really careful to say these things, but of course, if you're not trained with how scientific language works and what we mean by that, and the safety of an extrapolation, and the way that we can present risk and uncertainty, and 95% confidence intervals, complicated mathematical understandings of what the data means and how it's likely, it's very hard to explain that to 50 million people in the UK so that they're all comfortable with the science that's been done behind vaccine development. I don't think you'll ever succeed completely. I don't think we did ever succeed completely. Particularly at the beginning, we have no training in this, in speaking to the public about some of these complicated terms, and that the country, and indeed the world, was in a state of nervousness and fear and uncertainty. And so they wanted certainty from us. And I think that we're not always prepared to give that, because you can't.
No, yeah.
And the fact that we don't is then counted against us, because those who are, we would call them anti-vaxxers, deploying vaccine disinformation, give certainty. And I still see this. There's like on Twitter yesterday, there's still some, I'll say bullshit because it's bullshit circulation about adverse events post-vaccination. That it's data which is being entirely misinterpreted by malicious players, but the data is true. They circulate true data with an incorrect conclusion. And the reason that, and you have to debunk it by actually getting into the statistical reasoning as to why the way that you are interpreting this data is not the actual true result. And it's very complicated to debunk those, and so we lose that war, because we have to represent the truth as it is the truth, and we can't just say black and white, this is fine. You have to say this is fine under these circumstances, and from that we expect it to be fine under those circumstances, but that is an expectation. And it's COVID, it's hard. Very you said the word frustrating. It's very frustrating. But we have to get better at telling the facts correctly, but in a more story format. And that's, I think, why we tried to write the book in a kind of a story format, try to explain that the science is good, it's being done by good people with good intentions, and the complexities of actually having to tell the truth accurately. We can't lie, whereas the misinformation side can just lie, and that's very hard to. And
also COVID is very perfect, as in like you can't see it. And I was always, when I was explaining, I was showing pictures of measles and variola, and I was like, if COVID looked like this, how long would it take you to get anyone you love to get a vaccine? But the COVID, because it's so invisible and it looks like a cough, but it doesn't, it's not it's
just the flu. It's like, it's not just the flu.
Exactly.
Speak to anybody who was working in ICU in March of 2020 and tell them COVID is just the flu. I mean, we saw our colleagues in the healthcare sector broken by the awfulness of this disease, and yet nobody I knew got sick. Anecdotalists. Oh, like, well, you probably haven't got many friends, mate. But it was just awful. And also there was, I think, this prejudice that it was mostly affecting older and vulnerable people. And we saw that in our politics, and we saw that it's just people who would die anyway with some of the awful things that were said. Shocking. And anybody who was in healthcare would stand up and say, no, this is not okay. And that's right. Imagine if it had been a disease that predominantly affected children. Would have been a completely different response. The messaging wasn't always right, but everybody was afraid, and people like certainty. I think feels to me that we wanted certainty. And although as scientists, we're trying our best to give you the best information that we have, one thing scientists never have is certainty, because that's the whole point of the game.
Yeah, and it's interesting because even some scientists thinks that science is about certainty, and I was also seeing kind of predictive models of how things would be, and I'm like, but you guys, they just don't have enough data to speak about this. And I guess there's this idea of pattern needs to be given time to emerge. And I think even as scientists, we forget that sometimes. I guess because we are in it for a long time, there are a lot of certainties now. There are truths, we know our truths, but those took 100 years to get to. It's
and that was hugely challenging for all of us working on vaccines or treatments or anything. That 2020, trying to tackle a new disease when you can only go on assumptions. I mean, some very valid assumptions and some well-grounded assumptions based on, decades of experience with other respiratory infections. There are experts that could give the best models we can have. But we were always working in this against this background of this is a new thing, and we don't know if any of the things that we're trying are going to work. And so throughout the clinical trial, so we have our first human immunology data by about June 2020, so we could see that we were getting the right kind of antibody responses to the vaccination in our first phase 1 trial participants, and we could see that we were getting T cells, the right T cells that we'd expect. So every indication by the summer that the vaccine would work, but we have no correlate of protection at that stage. So we don't know how much of an antibody response you need to be protected. Perfectly possible you can make detectable antibodies, but that not be a protective effect. So in order to get to the idea that this vaccine is going to be effective, you have to wait until your phase 2 efficacy trials are in, where you've got a placebo group and a vaccinated group going out in the world, and some of them being exposed to COVID and therefore some of them getting sick, and you have to collate enough events, which in our case was hospitalizations with COVID, in that trial cohort, and then unblind it and see whether you get more or less hospitalizations in the vaccinated half of the trial. And we didn't get that data till the end of November. So it took that entire of 2020 to be able to amass data to be sure that the vaccine that we had made is protecting people from what is the serious side of COVID, which is getting hospitalized with COVID. And so we didn't, all of that talk we were doing, all of that, public facing events we were doing throughout 2020, Sarah's never able to say we've got a vaccine that's going to work, because we didn't know that until really the first week of December.
And actually, I'm also curious about the book. So in this journey, you have become a person that makes vaccines, that speaks to the public, and is a writer. And how did the book come to be? And what was for you the purpose of doing it?
So in the summer of 2020, I'm a fellow of an Oxford college, so that's a community of academics that, as across subjects, that get together to teach undergraduates mostly, and it's also where our undergraduates live, so it's kind of like a hall of residence for other universities, but with academics associated with it. And obviously in 2020, all of our social life was off. There's no dinners, there's no meeting with colleagues, there's no having a cup of coffee and saying what's going on with your research. So we were doing these Zoom after dinner get-togethers where one of the academics tells a bit about their research or their presentation. They're really interesting. I've got colleagues that work on medieval Spanish or on macroeconomics or on geology of ice flows in glaciers in Iceland. Really interesting, diverse bunch of academics. One of the joys of being an academic at Oxford, actually, really broad area of expertise that we can tap into. So I just did. They asked me, they heard I was doing some vaccine stuff, and they'd asked me to give a talk on Zoom after dinner with a glass of wine, tell everyone what's going on with the vaccine, because people are interested. So of course I'll do that. And randomly in the audience of that talk was an alumnus who is a literary agent. And after I'd given the talk, he hounded me for like 6 weeks. And Neil, he would agree. They pretty much just emailed me every day saying, "Kath, that is a brilliant story. You have to turn that into a book. You've got, there's the world needs to hear this as a story, as in long form, because we were doing quite a lot of press, but it was all short form newspaper articles. And so to get it all in context, Neil had felt that there would be an audience for that. And I, of course, said, well, I can't do this by myself. It's not my story to tell. So I had emailed Sarah and said, look, I've had this interest from a guy who's legitimate because I met him through college. And I expected Sarah to say no, she's mad busy by June. So everything's chaos. I expected to say, there's no way we're going to write a book, Kath. And she set up this meeting with Neil, and Neil's very charming. He's an agent, so he's charming. And he sold it to us as the, we will set you up with a fantastic, a writer who will work with you. So there will be weekly meetings where you either send her drafts or you send her oral recordings, and she will take that and make it into you don't have to do the physical writing of the book, though I actually think by writing as well. So I did just draft all my sections and then emailed them to her. But Sarah, I think, did a lot speaking them. So Deborah was our writer editor. She worked with us. So the three of us, Deborah, me and Sarah, just sat through for the last half of 2020, and just, we were then doing it contemporaneously, writing down what was happening to us, filling in the sections that we thought would make it into a round story. And so because we were doing it in real time, it wasn't that hard to do. It's not a hard book to write, because you're just writing down things that are actually happening.
It's like a diary almost.
yeah. So we were doing it as we were going, with a delivery that we would get that delivered by March 21, and it was published in the summer. So but then how, the again, you need an expert. Sarah and I don't know how to write a book, but because we've got this agent on board, he knows how to go out and find a publisher, do all of that, sort out so we didn't have to do any of it. We just had to deliver. I had to deliver a chapter every 2 weeks, was just writing down my thoughts and views and opinions and things that I had done. And then a lot of redrafting, because Deborah was very good at, as you can see, I have very long sentences. I just keep going, stream of consciousness. And Deborah was very good at saying, right, I've taken that enormous paragraph and I've cut it down to these 2 sentences, Kath. Please check that those are the 2 sentences that best represent your enormous, florid paragraph. And she was very good. And then a very intense editing, because for Sarah and I, it was really clear that we needed to be concise, but absolutely precise. If we're going to use an analogy, it has to be really watertight. It has to be really not dumbing down the science in any way. We want people to be able, because the science isn't complicated, so you should be able to explain it to your nan what a replication incompetent adenovirus vectored vaccine is. And I hope we did that. And also to get across some of our personality and get across some of the fact that we're not evil core crazy men in white suits setting out to put 5G chips in the world for our own personal gain, that's not our story. That isn't the motivation, and it isn't what happened. And I hope that we got some of that across. It was really well received, the book, so I hope that we did all right.
Yeah, I mean, I personally thoroughly enjoyed it, and I thought, I mean, there is a part of me, that having 2 women write this and lead this whole project, I was like, yes, role models, thank you. So that was also a part. And I actually, maybe one of the last question is, how difficult was for you to be a woman, or easy to be a woman in science?
So, I mean, Sarah always laughs at this question. I mean, I have no idea. I have no other option.
Well,
so we can't address that. And if you look around our teams now, and this must be the same true for you too, that most of the people that do science in my field are women.
That's a true.
Postdocs are heavily slanted these days to being female, and this is not yet true at the PI level, which is very frustrating.
That's it.
It's quite clear to me why that is, and that's because the structural complexities of trying to have children in this crazy country where childcare is, $2,000 a month or however much it costs you, and your housing is already half of your salary, and the postdoc salaries are not high. So how the hell are you expected to raise children and still be in this highly competitive working environment where every single year you have to produce something, because that is how we demonstrate that we're still active in science? It's extremely challenging, I think, to be looking at being a family, the main caregiver in a family, and also being a scientist. I think we have to fix that. It's a structural, and it's just clear that both the it's the measurement of success has to alter to take into account significant caring responsibilities, and that there needs to be better maternity arrangements for postdocs, because often you're on fixed term contracts that then basically don't have maternity cover if your maternity cover stops in the middle when your contract is up. I mean, I have to fight all the time for women in their 30s in my team who want to start families, and there's often very limited support for them at any stage. It'd be much better to have your kids when you're 24 and then start a scientific career, and that's obviously not feasible these days. So that was a huge challenge for me. I have one daughter. She's 12 now. And for me, maternity was really difficult. It came at the point where I just got a career development fellowship. So I'm trying to start a lab, but I'm also trying to start a family. It was hugely challenging for me, and was a very difficult part of my life, trying to juggle all of that. I was very stressed for many years. It was really difficult. I didn't feel supported by a lot of the institutions around me. It may be slightly better now because that was 12 years ago, but it was really hard. So I would like to see us do much better on that over the next 5-10 years. I think some policies have improved around maternity leave, but not around the fact that when you take a career break, you by default don't write any papers that year.
Yeah, and what I guess I'm curious is who are your allies in that fight, and who do you fight allyship with?
Yeah, well, I felt very unsupported, and I didn't find allies during that particular period. It was really difficult for me, and I just had to draw on internal strength and a FU attitude, like, damn it, I'm just going to do this and I'm going to have to plow through it. But it was not good for my mental health. It was not good, and it was really challenging for my family. So I obviously, the solution to that is a general society-wide solution, is that if we can get men and women sharing equally all of the aspects of caregiving, whether that's caring for elderly parents or caring for children or caring for a pregnant or postpartum wife, if we could really embed that as being not a job only for women, and it's obviously not a job for only for women, and we start to see moves towards understanding that men could do that job just as well, the same way as women can be scientists, men can be caregivers. If we could get to an equal society, all of this goes away, because having a child would impact on both genders equally, and then there would be no difference. But that's obviously not something we've yet, and I would really like to see better parental fatherhood provisions, because I think that that's the way we make things equal, and equal to the best for both genders rather than to the worst for both genders. We don't we should make, and I still see that. I see that in my teams. I see young fathers who struggle to shared paternity cover is a mess. You try and get two institutions, because they don't work in the same institute yet. You've got to try and get two companies to work together in order to figure out how to do shared paternity cover. It's a mess. I would really like to see it fixed, and society to share equally these roles and all of the other roles involved in organising the house. But both Tess and Sarah and we are all mothers, and we all managed it, and it comes with great stress, but we all manage it. Nobody ever asks the men on the team if they're fathers. I think that's because they know that traditionally that hasn't been such a hard thing to do. Juggle fatherhood with professorship. It seems to still not be as hard. I would like to see that change.
And I guess what I am very touched by is the fact that you're not like, well, it was really hard for me, so it has to be hard for you.
Oh God, no. It really should not be. It's not right. It's a great loss of talent. I see a lot of postdocs who come and who then get pregnant during their postdoc, and then they kind of drop out of the academia because they see that the route actually through to becoming an industry scientist is going to be more sensible for managing a scientific. You can still say a scientist. Many postdocs stay scientists, but will go and work for interesting biotech, interesting startup. Just choose not to continue their scientific career in academia because the perhaps additional pressures of academia, feeling like it's a job that is 24 hours. And it's not achievable. You can't be at work 24 hours. I don't work weekends. I do not check my emails at weekends if I can absolutely avoid it. I mean, I might flick them, but I won't reply. I can't. I have to have time. I have to have time with my daughter. I can't possibly. It's not necessary for me to be at work. I mean, during COVID, we work weekends.
But
that was a special time. And there's a special thank you to my daughter in the book, because she knows that her mum was not around for her in 2020 as much as she wanted to be. Yeah. And that I had to repay her with a very fancy holiday to Japan this year. There was always going to be the thank you to Ellie for being brilliant during COVID.
And actually, it's interesting how you're speaking about this. Is almost as if you're apologetic of like non-working weekends, as if that is expectation. And I think that's an incredible
madness,
ingrained madness. Exactly, especially because science is a job, and we always forget that idea. I guess because of the passion with which we do it, but it's still a job. To round off and be respectful of your time, the last question is, I like it because of the idiosyncrasies, but is there an absurd thing about you that not many people know about, and they would not connect to you, but it's what you are and who you are?
Well, people perhaps know about it more now because I've done a few of these and we wrote the book. So my secret passion is like going out raving. So I'm an old school dance music enthusiast, and I like nothing more than going to a rave in a field with a lot of strangers and putting my hands in the air and having a dance. It was one of the things I missed most in COVID, was the ability to socialize with others. So I like to go clubbing.
And what kind of music do you like?
Oh, absolutely anything. I was brought up on kind of like indie rock and roll, but then during a postdoc in Brighton, I got much more into like the club scene in Brighton. So I have a good friend, Bernie, and we used to go out to house nights together. And now my crew are quite diverse. I have a lot of non-scientist friends, and we'll go and listen to anything from reggae to dub to ska to Afrobeat to anything live or recorded. If it involves dancing in a sweaty room with some strangers, then I'm down for it.
Lovely. This is my favorite question. So thank you very much. Is there anything that I haven't asked you, but you have a burning desire to share?
There is not. That is my last question that I always ask at interview. So it's a good question, but I think that's been a fairly wide-ranging conversation. I hope I haven't given too much of myself away.
Well, I hope you did, because I think it's so important. Okay, so thank you so much for your time and for being willing to join me in this.
Thank you. It was very nice to meet you. Have a good day.
You have just heard the story of Catherine Green, an English biologist who is an associate professor in Chromosome Dynamics at the Wellcome Centre for Human Genetics at the University of Oxford. Her research is dealing with chromosome stability during the replication of DNA. During the COVID-19 pandemic, Green was part of the Oxford team who developed the Oxford AstraZeneca COVID-19 vaccine. She is also one of the co-authors of the book Waxers. Thank you for joining us on this journey. And if you liked it, please share and subscribe so that these stories can reach more people.